Hello, and welcome to today's presentation, How do we translate clinical and preclinical research into labeling? My name is Suzanne Minton, and I'm a scientific content specialist at Certara. Before we begin, I will cover a few housekeeping items. At the bottom of your audience console are multiple application widgets you can use. If you have any questions during the webcast, you can click on the Q&A widget at the bottom left and submit your question. We will try to answer these during the webcast, but if a fuller answer is needed or we run out of time, it will be answered later via email. We do capture all questions. The slide widget is next to the Q&A widget. You can expand your slide area by clicking on the maximize icon on the top right of the slide area or by dragging the bottom right corner of the slide area. A copy of today's slide deck and a link to an on-demand version of this webinar will be emailed to you approximately 1 day after the webinar. The on-demand version of the webcast can be accessed using the same audience link that was sent to you earlier. Next to the Q&A widget is the Help widget. It has a question mark icon and covers common technical issues. Often, simply refreshing your browser will fix most technical problems. The next icon looks like a green folder. This is the resource list where you can find additional helpful materials, including the registration link for the next webinar in the series. Next, there's a purple Contact Us icon, which you may use to contact us regarding any of Certara's products and services. Before we begin the presentation, I would like to introduce today's speaker, Dr. Karen Rowland Yeo. A full speaker bio can be found in the blue widget at the bottom right of your console. Dr. Karen Rowland Yeo is currently a senior scientific advisor and principal consultant. She supervises a team of scientists who extrapolate in vitro data to predict in vivo pharmacokinetics in humans. She has helped develop and implement models within the Simcyp simulator. Karen has also been heavily involved in consultancy projects related to the application of the Simcyp simulator within the pharmaceutical industry. Karen received her bachelor's degree in physics from the University of Natal and a PhD in drug metabolism. She then went on to complete a postdoctoral position in childhood leukemia and a lectureship in clinical pharmacology and therapeutics at the University of Sheffield. She has published more than 60 peer-reviewed journal articles. Karen, welcome to today's event. I'll now turn it over to her to begin today's presentation.
Thank you very much, Suzanne, for that kind introduction. Okay, so as Suzanne's indicated, what I'm going to be talking about today is how do we translate preclinical and clinical research into labeling. Firstly, what I'm going to cover is I'm going to give an introduction to PBPK modeling. I know that many of you will be familiar with this, and therefore this will be repetitive, but also some of you will be less familiar, and therefore I think it's important to cover the system and drug-related parameters and also talk briefly about how we integrate inter-individual variability. I'll obviously be covering some of the labeling examples that include PBPK modeling, and also talking about this with reference to some case studies and giving an indication of the relevant in vitro and clinical data, and also talking about the model development side of things. So why are we interested in PBPK modeling? Essentially, if you have a look at this figure on the screen here, you can see that on the left we have the systems data that consists of age, weight, tissue volumes, obviously many more parameters than this, but they're basically the physiological data and also the data they use to drive some of the models. Also in the middle you can see the drug-related data, physicochemical parameters, also metabolic, permeability, transporter data, solubility data. And on the right-hand side, you can see that we have the trial design aspects. All of these can be combined, and then what we can do is use the in vitro data, extrapolate it to in vivo data or in vivo estimates using these systems parameters. We integrate this within a PBPK model and also indicate what the trial design conditions are, and what we can do is extrapolate or predict to the drug PK or PD in the population of interest. And essentially that's why we're interested in looking at PBPK modeling, because once we've input this data, then by changing the systems data or anything that affects the drug data, we can see what overall effect it has on the PK exposure in the population of interest. So what I'm going to just show you now is just that obviously in terms of the model selection, what we can do is we can use a lumped minimal model that's got a reduced number of compartments shown here, Or on the right-hand side, if you're interested in looking at the exposure in all the different tissues, what we can do is actually use a full PBPK model. And then this is obviously, we would need all of the parameters actually describing all the different compartments and the flows. In terms of the absorption side of things, we can simply assume first-order absorption, or what we can do is apply a more mechanistic model, and in this particular case, it would be the ADAM model, which is the Advanced Dissolution, Absorption, and Metabolism module. If you're interested in having a look at the regional distribution of 3A4 and PGP along the intestinal compartments, or simply wanted to have a look at the formulation side of things. But obviously, using these more complex models, you do need more data to drive them, and therefore it's very important at the start of the project to decide what is the question you're trying to answer before you actually go on and have a look at the model selection. So one of the things that we've been very interested in from the start at SimCip is having a look at the challenge of population variability. Often when drugs are taken off the market, it's because of an extreme situation and not based on the actual average man. So if we have a look at these aspects here, what we're interested in doing is having a look at the impact of environment, which could include drink, which may lead to cirrhosis or smoking, or in actual fact disease or genetics. I always joke that this looks like me in the morning before I've had my first cup of coffee, and this is probably me at the end of the day by the time I've had 10. But anyway, what we're interested in doing in SimCip is actually generating the population variability. So how do we do that? Well, within SimCip, what we do is the first thing is that we look at the target population that we're interested in. It could be healthy volunteers. It could be an oncology population. And what we would do is actually derive the age-sex distribution for that population. We would generate a height from an age, a weight from a height, calculate the body surface area, and obviously then what we would do is calculate other physiological parameters that were related to body surface area and try and build all of these covariates within the framework so that we can generate individuals who are representative of the actual population. So within SimCip as well, obviously, apart from those covariates, what we do is we try and derive distributions for organ tissue distributions, the different sizes here, also the enzymology as well, what is the variability associated with CYP3A4 expression, and all of these data sets we collate from the literature. And then what we would do is integrate these within the SimCip simulator and then use these to actually help build the virtual population to actually predict the variability within a full population. So if we move on to this next figure here, what we're looking at is Monte Carlo versus correlated Monte Carlo approach. Obviously, I've talked about the fact that we sample from distributions to put a virtual individual together, but what we want to make sure is that the organ sizes are relevant to that individual. Quite clearly here, you can see that these organs were randomly generated using a Monte Carlo approach and assigned to the individual, but the sizing isn't correct. However, in this individual on the right-hand side, you can see that all of the organs fit appropriately in adult and also a pediatric subject. And this is what we mean by a correlated Monte Carlo approach. So even though we're actually sampling from these distributions, where possible, we're trying to correlate these with age or body surface area so that we end up with appropriately sized individuals. So now that I've given you a bit of the background, what I wanted to show you here is go back to this slide where we're talking about the systems data. So what you've done is you've incorporated your drug data and then simulated that out, and then perhaps actually got some PK data in healthy volunteer. But now what happens if you want to project that to exposure in a cirrhotic population? What would happen is we would load up the appropriate parameters here for cirrhosis population and apply that to your drug compound file and see how that impacts the PK parameters. And same with an obese population, differences, Also, you can see an elderly population and also pediatric population. So just to explain again, and I'll give an example of this is just integrating the drug data here, applying the relevant system parameters, and then looking at the impact in the population of interest. And what I'm going to be showing you later is a development of an oncology drug where it starts off in a healthy volunteer population, how we change the parameters relating to the oncology population and see what impact that has. Now that I've given you a bit of a background on PBPK, what I'm going to be showing you now are some of the label approvals that have occurred over the past few years. And on this slide, what I'm showing you here are the drug names, the company names, and also the therapeutic indication on this side here. In terms of the, on this column here, what I've got are the studies cited. Now, when we started off doing this, I tried to have a look at studies avoided, but that's quite difficult and that's quite subjective. And also, it's quite clear that even in some of the labels, for some of the example, PBPK modeling is mentioned, but for some of them it isn't. But when you actually go back and have a look at the relevant reviews that have been conducted by the FDA, the PBPK modeling has been mentioned, and that is why all of these examples are here. And so just to give you an example here, and I'll be expanding on this, is in the case of ibrutinib, what I'm saying here is that PBPK modeling has been referenced against more than 12 clinical studies, and it's the same here. They may not have all appeared in the label, but certainly in the review, PBPK modeling has been used to inform dose selection or study design or other aspects, and I'm going to come on to that more in the next few slides. Before I go on to that, one of the examples that I want to highlight is cimeprevir, a Hep C drug. And even though for this particular drug, when you actually have a look at the label and the associated review, what you see is that more than 20 clinical DDI studies were actually conducted. And at this point you say, well, what is the point of developing a PBPK model? Well, the point about it is that these drugs are actually very complex. There are a lot of transporters involved, a lot of different enzymes, a lot of things happening in there. And therefore, all of these DDI studies were conducted, but in the actual review, PBPK modeling was used. It was validated using or verified using all of the clinical data, but then it was actually applied to look at to try and describe the nonlinearity of the drug across different populations as well. And it was shown quite clearly then that the nonlinearity occurred because of saturation of transporters and metabolism. So again, just to indicate that even though many clinical studies can be performed, there is always a place for PBPK modeling. So what I'm going to do now is show you that this is an example of liglistat, and this is the actual wording that was taken from the PBPK label, and you can see here that co-administration with CYP2D6 inhibitors, and what it does is it describes the clinical study that was done, and then it describes the PBPK models and how they were used to indicate how the PK of the liglistat may change in CYP2D6 EMs and IMs. And here we've got the information regarding CYP3A4 inhibitors, and here simulations were run where both CYP2D6 and 3A4 inhibitors were actually applied. So here we have 12 DDIs that were actually predicted in CYP2D6 EMs, IMs, and PMs, and that's quite a significant number. And all of the wording appeared in the labeling relating to these particular drugs. Now, if we go on and consider this, what information would we actually need to get to that stage? If you think about this drug, it's mainly metabolized by CYP2D6, also 3A4, and there may be a minor renal clearance component. It was indicated in the review and labeled that there were clinical DDI studies for paroxetine and ketoconazole in CYP2D6 EM subjects. This is good because what it means is that once you've developed the PBPK model, this can be verified using clinical data, and obviously to get the CYP3A4 and CYP2D6 contributions correct, because that's what we need to do at the end of the day when we're looking at a DDI, make sure that we get the absolute enzyme contributions correct. Also, what we have to consider as well is that we need the different CYP2D6 abundance data for the different phenotypes, for the EM, IM, and PM subjects, to incorporate that, because the susceptibility will be different for the different phenotypes. And once you've gone through all of that, then you can use a verified model to predict the DDIs for the different phenotypes. And in fact, it looks like that is what was done. But then the thing about it is, obviously what we're interested in is then using that information and the simulations to provide dose recommendations in CYP2D6 EM subjects. And you can see here, this was taken directly from the label in the review. This is a recommended dosage by CYP2D6 metabolizer status, so in EMs and IMs. And you can see here that in the case of strong or moderate CYP2D6 inhibitors, given concomitantly with strong or moderate CYP3A inhibitors, these are contraindicated because obviously at this point you're knocking out most of the 2D6 and also the 3A4, and therefore the DDI potential is great. Similarly, in the case of paroxetine, you can see that with EMs and IMs, the dose has been reduced to 84 mg once daily. The normal recommendation for dose would be 84 mg twice daily, and you can see that they've recommended a dose reduction in this particular case. And most of these data, there was obviously a clinical data to support this one, but in the other cases, it's based on predicted data. And this is actually shown here based on drug interaction studies or predicted interaction in EM and IM subjects. So there was one clinical study, and the rest of it was based on predicted data using the developed PBPK model. Also, dosage recommendations were provided for CYP2D6 PM subjects, and you can see in this particular case that CYP3A4 inhibitors are contraindicated and the moderate and weak are not recommended. And again, these were all based on predicted interactions as PMs as cited. So what I'm going to show you here is just a similar case study for Aripiprazole. This is a drug that's metabolized by 2D6, and it's also a 3A4 substrate. And what I'm going to do is just take you through the process just to show you when you would consider a model to be actually validated to go on and predict to unknown scenarios. So we would go through the model development, collate all of the in vitro data for Aripiprazole and also relevant clinical data. That may be single ascending dose or multiple ascending dose data. Then we would go through a model verification process. In this particular case, there were two clinical DDI studies that are available in CYP2D6 EM subjects, one a CYP3A4 inhibitor and one a 2D6 inhibitor. And obviously that allows us then to actually validate the relative contributions of the enzymes. Also, what we can do is look at the exposure of the drug in 2D6 EM, IM, PM subjects. And once we've got to that stage, once we've validated the model, we can then apply it to untested clinical DDI scenarios. And that's exactly similar to the previous example that I just showed, the oligostat. So in this particular case here, you can see the clinical DDI study with ketoconazole in CYP2D6 EM subjects. On here, we've got the control situation following a single oral dose of aripiprazole, and in this situation here, we get an increase in exposure when it's co-administered on day 2 of 15 days dosing with ketoconazole. And the predicted AUC ratio is 1.6, and the observed AUC ratio, again, I mean the two are consistent, which is exactly what we want. So that is with respect to the CYP3A4 inhibition. Obviously, the next scenario would be the CYP2D6, which would be the quinidine. Again, we've got the control data here, which is the aripiprazole given on its own, and then this is when it's actually co-administered with quinidine. Now, you can see here that the predicted AUC ratio is 1.7 and the observed is 2.1, but that is for the population as a whole. When you look at individual trials, we do get some that are about 1.9, which are actually closer to the observed data. Importantly, the next slide here, what we're interested in doing is we have to make sure that we can recover the exposure in CYP2D6 PM subjects. And here you can see this is a 15 mg oral dose, and certainly in terms of the exposure and looking at the clearance, the values are consistent. So far what we've done is we've looked at the relative contributions of 2D6 and 3A4, and what we need to do now is confirm the relative differences in exposure in CYP2D6 EMs, IMs, and PMs, because really this is what we're interested in. often we may have a clinical study in a 2D6 EM, but what we want to do is maybe project to IMs and PMs and avoid those more complex clinical studies where we have to do the intense screening and genotyping to ensure that we get these individuals. So here you can see that these are just the different trials for EMs, IMs, and PM subjects. So what I did was run simulations for Aripiprazole, but actually correct for the abundance data for EMs, IMs, and PMs, and then project out to a steady state concentration. And you can see here that these are reasonably consistent, and what I'm showing here is the exposure in IMs relative to EMs and PMs relative to EMs. So what I've done here is essentially collect the in vitro data for Aripiprazole, combine that with the clinical data, simulated out 2 DDIs and looked at the relative exposure. And at this point, I would be happy that the model is validated or verified and therefore can be used to extrapolate to unknown scenarios, exactly as in the Oligostat example that I've just shown you. So that's exactly how we would go about it. What I'm going to do now is present on ibrutinib. I know that many of you will have seen this. This example has been shown over the past couple of years. But what I want to do is actually highlight a few of the aspects that I found in the review that I think are quite important, because it's not only about the cited DDIs, it's about the other applications of the model as well. So obviously we know that ibrutinib is a 3A4 substrate. It's got a high clearance, and therefore we know that it's, just even looking at that, we know that the susceptibility to drug-drug interaction is high, even before you actually go on and have a look at the DDIs. So again, in terms of the model development, we have the in vitro data, or that was collated, and also the clinical data. And then a model was developed based on those actual data, and this is the actual strategy that was presented in the review. And then, of course, the model was verified using 2 clinical DDI studies, Ketoconazole and rifampin. And then, of course, it was applied to untested scenarios, in that case, moderate CYP3A4 inhibitors and inducers. So, of course, this figure, I think, as well, which I'm going to show now, many of you are familiar with. And what it's showing is the ketoconazole observed, ketoconazole simulated, erythromycin, diltiazem. So all of the different inhibitors, starting with ketoconazole up here as a strong inhibitor and rifampin here as a strong inducer. The observed data are shown by these turquoise solid blocks, and these are the corresponding simulated data, and exactly the same with rifampin. And what you can see here is this is just actually showing you that the model, the PBPK model that was developed, was able to recover the observed data with respect to ketoconazole inhibition and also with respect to rifampin introduction. And of course, at that point, it was verified. The model was it was reasonably robust, and therefore the model was then applied to these untested scenarios here, where they were looking at the impact of moderate and weak inhibitors and also a moderate inducer on the change in exposure. Now, of course, this was the wording that then went into the label. Co-administration of ibrutinib with CYP3A4 inhibitors. Simulations using PBPK models suggest that moderate CYP3A4 inhibitors, CYP3A inhibitors may increase the AUC of ibrutinib 6 to 9-fold in a fasted condition. So this was the precise wording that actually went in there, and also again mentioning the effect of a moderate CYP3A inducer. Now, obviously, this is quite important, but as I said, if you just look at the label, then you think, right, okay, well, the model was developed and verified and applied to these untested scenarios. But really, I think, you know, the model was used much more than that, and that has actually come through in the review. So what I'm going to show now is, at the end of the day, what we're interested in is, yes, we want to know what the DDI susceptibility is, but really what we need to be able to provide is a dose optimization strategy. So if we have a look at this slide here, so for one of the therapeutic indications, the recommended dose is 560 mg, which consists of 4 140 mg tablets QD. So that is the recommended dose. But obviously, when you're thinking about CYP3A4 inhibition, what we have to be thinking about is a safety issue, because obviously we're going to increase the plasma concentration of that drug by quite a lot, and therefore we need to know what dose we can actually give in the case of co-administration of CYP3A4 inhibitors, so that we don't go beyond that. Some of the highest doses that were evaluated were about 840 to 1400 mg, and therefore safety data will have been available up to here. And obviously, one of the lowest doses that is recommended is 140 mg, because that represents really the efficacy side, where 90% of the relevant receptors are occupied. So the recommendation here was that if you think back to the previous slide, in the case of moderate inhibitors, there was a 6 to 9-fold increase in exposure that was predicted. So the recommendation was to reduce the Imbruvica dose to 140 mg in the presence of a CYP3A4 inhibitors. So if we actually think, right, this is actually going to be the recommended dose, and if you've got a 6 to 10-fold, you're still within this safety margin here. And that is why this dose recommendation was actually mentioned and suggested. And the reason I mention this is that at the MHRA meeting last year, and I think also maybe at the FDA workshop earlier in the year, one of the things that was mentioned was, yes, I think what a lot of people are doing is they're assessing the DDI potential, but then what they're not doing is extrapolating to dose recommendation. And this is just to show you how the dose recommendation was made. Obviously, in the case of CYP3A4 inducers, what we're concerned about is this, again, as a daily dose, is that in CYP induction, where you get an increase in clearance, it may be that this level is going to drop below the exposures that are actually projected from a 146 milligram dose. And therefore, what we have to do is try and come up with an increased dose, whereby when you introduce a CYP3A inducer, you're not going to end up with too low a concentration. And this is actually just a table showing simulations, I think, that were done showing here you've got the interacting drugs. So you've got efavirenz, which is a moderate CYP3A4 inducer, and you have rifampicin. And in this particular case, at the bottom here, what was done was the ibrutinib exposure ratio is presented with an inducer relative to the 146 mg dose with no induction. And what they did here was, is the exposure ratio versus 140 mg above 1. So in other words, what they're saying is, if you give 560 mg of the ibrutinib with efavirenz, do you still end up with an exposure greater than 140, which is obviously the efficacy, dependent on efficacy data here. And what they've said is that with a moderate inducer, in the case of efavirenz, yes, it is. But in the case of rifampin, even if you double the dose to 1,120 milligrams, you're still not going to get a higher concentration than the 140 milligram dose. And therefore, in this particular case, it states avoid concomitant use of strong CYP3A4 inhibitors, and that's based on this data here. So that is something, as I said, that we I think have to get used to, is that yes, even though we may assess the DDI potential, what we have to think about are the safety and efficacy margins to be able to extrapolate to a dose recommendation in the label. Another interesting aspect as well where the model was applied was the effect of dose staggering. And of course, this is very, very important, especially for a drug like ibrutinib that is high extraction, because it's susceptible to first pass, a lot of first pass inhibition, and therefore dose staggering could have quite a large impact. And in actual fact, what you can see here is in this table, you've got CYP3A4 inhibitor in the dosage regimen, the inhibition mechanism, so you've got time-dependent inhibition down here, and we have reversible inhibition here. And you can see that this is actually scenario 1, whereby the 2 hours before the inhibitor, and the ibrutinib is given 2 hours before the inhibitor, and here it's actually co-administered. So, of course, this is the clinical situation here, but in actual fact, you can see that just by having that dose stagger between the ibrutinib and the ketoconazole, you can end up with a very, very different result. And even though, obviously, this hasn't gone into the label, it is still very important information to know, because I mean, obviously, if individuals take the drugs, with a lag in between them, then you could end up with a very, very different result. But obviously, this is the worst case scenario here, and that can be shown on this next slide here. If we have a look at the enzyme activity after multiple doses of competitive inhibitor, you can see that this is a change in the ketoconazole here with time. On the right-hand axis here, what we've got is the corresponding change in enzyme activity. So obviously, as your ketoconazole concentration is high, your enzyme activity is low because you've got all of this inhibition here. And therefore, it's very important to see or to have a look at the impact of when you're giving your ibrutinib, and it's very dependent on obviously where the enzyme activity is relative to the time when you give your ibrutinib dosing. And another thing to mention at this point here as well is that simvastatin, obviously another high extraction drug, and that is susceptible to dose stagger as well. In fact, when you give ketoconazole concomitantly with it, you end up with about a 30 to 40-fold interaction. Even if you have a 1 to 2 hour dose stagger, that can be reduced down to about 10-fold. So it's not just with ibrutinib, but there are a number of other drugs as well. And as I said, it was important to be aware of that, even though it hasn't affected the label. So one of the other important aspects as well was that what I noticed is that in the review statement here where the PBPK modeling was reviewed, it says, we noticed that you use PBPK modeling to predict drug interactions. You should further investigate the potential PK differences between healthy subjects and oncology subjects. And I think this is very, very important because I think many, of the examples that I've seen so far, even though we're more interested in what happens in the patient population, a model is developed in the healthy volunteer situation and then not necessarily applied to the population of interest. And so this is just one thing that I picked up. And what I'm going to show you now is just an example that was developed in a healthy volunteer population, but then extrapolated to an oncology population, and just to see how we would actually handle that. So again, very important, this is often the question that I'm asked, what data are required to actually develop a PBPK model? Well, in this particular case, for this particular drug, there were metabolic data available for human liver microsomes, hepatocytes, and RCIPs. FM CYP3A4, quite a tight range, really. I mean, I'm saying 70 to 80, but this was, quite a robust value, and all of the numbers tied up in with respect to the recombinant CYP enzymes and also HLM and chemical inhibition. FU data were provided for a range of concentrations, physicochemical parameters, permeability data, and also there were inactivation data available for CYP3A4. But I wanted to make you aware here that both the parent and the primary metabolite were time-dependent inhibitors, and therefore both of them had to actually be built into the model. But essentially, this is the minimum data required to actually develop a PBPK model. Importantly, as I've indicated in the previous examples that have been cited in the label, clinical data were always available. And so therefore it's important to have a look at the clinical data to see what is available to help you develop the model, and also once you've developed that model, which independent data sets then you can actually use to validate or verify the model. And this is pretty standard, single ascending dose data, multiple ascending dose data, food effect data, and the pivotal DDI study whereby we can confirm the contribution of CYP3A4 to the metabolism. So at this point, obviously we need to build in the metabolite, and therefore this is the model that was used to actually develop or to build the PBPK model. You can see here we've got the parent drug and the different compartments here, and on the left-hand side here we've got the metabolite. Obviously, because the metabolite is formed by 3A4, it will be formed in both the gut and also the liver. And as soon as it's formed in the gut and the liver, it's also susceptible to metabolism as well. So what I did was use single ascending dose data to actually help validate the build the model for the parent and also the metabolite itself. And you can see here that these are just the exposures. And what I did was, I'm not going to show it because of the interest of time, so I use a single ascending dose data and in vitro data to help develop and build the model, and then I use the multiple ascending dose data to actually show that I could recover both parent and metabolite exposures over multiple dosing. And of course, as I mentioned, one of the aspects that I had to build into that was time-dependent inhibition. And of course, in the presence of a competitive inhibitor, what happens is the enzyme pool remains relatively constant because you always get release. The inhibitor can always be released, and same with the competing substrate. But in the presence of a time-dependent inhibitor or mechanism-based inhibitor, you get accelerated deactivation. And of course, in this particular case, we've got both the parent and the metabolite that are contributing, and therefore both of them have to be considered. But essentially, this is just to show the schematic to incorporate time-dependent inhibition. So at this point, as I've mentioned, we have a model that works for a single ascending dose and multiple ascending dose. But now what we want to do is use this model to see whether we can recover the DDI data with ketoconazole. And obviously this is a complex drug-drug interaction because what we're looking at is the autoinhibition on drug X and M1. We're also having a look at the effect of the metabolite on the parent's metabolism and the parent on the metabolite inhibition. But then obviously also what we're building into that is the competitive inhibition by ketoconazole on the formation of the metabolite and also the clearance of the metabolite itself. So this is what we had to simulate. So what I'm showing you now in the next slide is what we would call the model verification or the model validation step. And here is just an output that we would get from SimCip. You've got an AUC ratio here, and these are the different trial groups. And obviously, as I've mentioned, what we do is we generate the virtual individuals within SimCip. We would randomly assign them to different trials because what we want to see is what is the variability across the trials when we're simulating or predicting the AUC ratio. And this is the observed data here. And you can see that there's an approximate 4-fold increase in exposure of the parent when ketoconazole is co-administered. But one of the things that I want to mention here is that obviously you can see that this was done after a single dose of drug X. Now, the more appropriate study design to have done would have been to multiple dosing of drug X to steady state, and then of course what happens is you get autoinhibition of the enzyme, and therefore your FM CYP3A4 is reducing over time, and then you would give the ketoconazole. But in that particular case, this was a study that was done, and therefore that is what I had to simulate out. But obviously what I can do is that now I've verified the model, is rerun the simulation with a steady state dosing of the parent, and then give the ketoconazole. Now, what I want to show you here is obviously this is exactly the same as the Ceretinib example that was, and this appeared in the review, where Ceretinib is primarily metabolized by CYP3A4. It's a reversible and time-dependent inhibitor of CYP3A. And what happened is a clinical study was actually generated, but in this particular case, again, a single dose of Ceretinib was actually given alongside ketoconazole, and it was shown that there was a 2.9-fold increase. What happened is obviously simulations were performed to actually assess what the true impact would be if the drug had been dosed to steady state and then ketoconazole given at the same time. And what you can see is that obviously, as expected, there's a significant reduction in the interaction because, as I mentioned, you've got autoinhibition occurring during multiple dosing of Ceretinib, and then when you give the ketoconazole, it's less susceptible to interaction. And then, of course, once the model had been validated with Ceretinib, then, of course, scenarios were simulated using moderate CYP3A inhibitors and strong CYP3A inducers and moderate 3A inducers, and these are the results that are shown here. So this is just to indicate that this case study that I'm showing is just similar to an example that has been shown up in a label and a review recently. So now what I want to talk about is how do we actually go about applying this PBPK model to oncology patients, because everything I've talked about for drug X so far has been done in healthy volunteers. What we're really interested in is what happens in these oncology patients. And so, as I've said, what we would do is we've developed it in healthy volunteers, and now it's gone into oncology populations and what happens. So this is using data that were put together by Chita et al. In 2012 from Genentech. And you can see here that, again, if you remember from earlier on in the lecture, what I'm interested in doing is first of all, establishing the age-sex distribution for the population of interest. And in this particular case, you can see that it's highly skewed to the right. And what this means is that we'll end up with more older people than we would in a healthy volunteer population. And wherever we have the covariates in place within SimSlip and this relationship with age, that will propagate through to a change. And so you can see that we have an MPPGL age relationships. This is milligrams of microsomal protein per gram of liver. And what happens is we end up, because of these relationships with age, with generating older individuals, automatically we end up with about a 30 to 34% reduction in CYP3A4. And therefore we will end up with a reduced clearance of the compound simply by using this age-sex distribution for oncology population. Obviously, another thing that we have to consider as well is that for an oncology population, what we're interested in is also the protein binding. We know that for AAG levels, this is a healthy volunteer levels, but in a patient population, this is an acute phase protein, and the levels can actually be much, much higher. In fact, I'm saying here about 1.4, but I have seen levels up to about 3. And therefore, this can have a significant impact. And what this means that if your drug is bound to more to AAG than it is to albumin, then obviously because of the increasing protein levels in patients, you can get a reduction in the protein binding. Also, if you consider albumin in the other case, what happens here is that you get a reduction in the albumin levels, and this can actually translate to a change to an increase in the FU. And therefore, it's important to know whether your drug is mainly bound to AAG levels or albumin levels, because as you can see, that it can have quite a large impact on the protein binding. And also, it's a good thing if you actually have the AAG levels or albumin levels measured in your population that you're trying to actually simulate, because then obviously this could have an even larger impact on the FU than the values that we would have in the program. So once we've incorporated those changes, so what I did is I took the healthy volunteer model, I implemented the age-sex distribution for the oncology population and also changed the protein levels for the oncology population as well. And then what I did was simulate to a much higher dosage regimen that were being used in the oncology population. And what I'm showing you here is that obviously after multiple doses, this is drug X shown on the left-hand side, and this is the actual metabolite shown here. So I haven't changed any of these parameters, just extrapolated from the healthy volunteer model, implemented the system parameters for an oncology population, and shown that I can recover it. So this would be, and I've been able to do this for several doses, and so this would be the verification model in an oncology population. And what the FDA was specifically interested in the comment that they made with respect to the ibrutinib is, okay, so you've done that now, what actually happens to the DDI potential? And obviously, you can see here, because we are using much higher doses of the drug in an oncology population, what is happening is we are knocking out more of that enzyme. So in this particular case, you can see that it's at a much lower dose, and during multiple dosing, you can see that you're wiping out the enzyme activity. And when you give the ketoconazole, this is the change that you actually get. However, at a much higher dose that is being given in the oncology population, you can see that you're wiping out most of the 3A4, and therefore the DDI potential is actually much less. And therefore, this was the observed data, and this is actually the projected DDI at a much higher dose in patients. And this is just to show you really how we can actually go about, because obviously what we won't be able to do is do DDI studies in healthy volunteers and also oncology populations as well. And therefore, this is actually just to show you, for an example, if you want to actually go on and use it in a PBPK label, or it's going to come for review. So finally, you'll be pleased to hear that this is my last slide, really. What I wanted to show you is that it's possible to develop a robust PBPK model to accurately predict clinical exposure of a drug, but you have to have the relevant in vitro data and good in vitro data. Even though you may have some clinical data which can be used to develop the model, you're still trying to gauge the relative contributions of enzymes initially from in vitro data, and in fact also maybe transporter data as well. So good in vitro data and some clinical PK data to help develop the model. And then importantly, some clinical DDI data to verify the model. And that has been shown in each of the examples that I've shown you, and also the labeling examples as well. Very important, clinical data to validate or verify the model. And then, of course, as I've shown, this validated model can be applied to untested scenarios, for example, the effects of moderate CYP3A4 inhibitors and inducers. And of course, this can then actually lead the results from the PBPK models can then be used in lieu of actual clinical studies or can inform on dose adjustments exactly as I've shown. So I would like to finish off now by acknowledging the SimCip team. And also, I would like to thank you for your attention, and I would be very happy to take any questions relating to the presentation. Thank you very much.
Thank you, Karen, for that presentation. We'll now be taking questions from the audience. You can submit them through the Q&A panel. And I'll start off with our first question. Karen, in your view, would the regulatory agencies accept simulations of transporter-mediated interactions in lieu of a clinical DDI study?
I think one of the things that this was quite something that was discussed at the FDA workshop last year, and I think regarding transporters, it's still early days yet. I think there's certainly most of the examples that I've shown today and most of the examples in the labels relate to CYP3A4, and therefore I think there's that comfort factor. I think with transporters, there have been a lot of increasing number of publications over the years showing that where we're able to predict and where we aren't able to predict. So at this point, I would say no, but things are changing as the data become more robust, and we also get abundance data becoming available for transporters.
When looking at a specific population, such as oncology, which parameters do you have to change?
Okay, so what I just showed you in the presentation was that obviously just by having the appropriate age-sex distribution, that can propagate through to changes as well. And also it's important to consider protein binding. One of the things that I didn't show in the presentation, but I think is quite important, is obviously the impact of cytokines on changes in CYP metabolism. Therefore, I think that is something that we will have to think about. But also the transient changes as well, because obviously they're going to change with the disease progression, but then also change with respect to the treatment. So I think that's something that I didn't mention, but something that we are going to have to consider in future.
Do you know of any examples where SimCip was used to predict food effects?
Not specifically, you mean accepted in terms of the labels?
Yes.
Yes. No, I don't know. I don't know of any specific ones, but there are some examples where the pH has been changed to have a look at the impact of proton pump inhibitors.
This looks to be the last question from our audience. You've shown some examples where clinical data was used to help validate the PBPK models that have been developed. What is the point of developing the model in the first place if DDI studies are still required?
Well, at the end of the day, I mean, that was why I mentioned the Cimeprevir example right at the beginning, where more than 20 clinical studies were actually conducted. The clinical data were obviously derived and were used to help validate the model. But, certainly then the PBPK model was actually applied to different scenarios to try and understand some of the nonlinearity. And in fact, I've worked on a number of cases similar to that over the years, whereby there has been a lot of clinical data, but it's still not been possible to understand the mechanisms, and therefore a PBPK has been used in those instances.
Okay, thank you. Our next webinar in the series will take place on April 8th. Doctors Bill Poland and Jason Perrone will be giving a presentation on population modeling of modified risk tobacco products. Again, you can register for this webinar and others in the series from the link provided in the resources widget at the bottom of your screen or by visiting Certara.com. At the end of this webinar, a short survey will be launched to collect your valued feedback. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.